All right. Thank you.
Aha, a crack.
Tell us about this crack, Adam. What caused it?
Movement of the ground. uh, hard to say. Yeah, though a lot of times these seamounts build up by adding volcanic material to the seafloor to the point where they kind of become gravitationally unstable and want to break apart a little bit. So could be that seamount grew too tall and starting to collapse a little bit, just to relieve that stress that has built up from the pile of material. Or it could be the weight of all these sponges.
That's a lot of them there, a lot of balisoma. What's that in the center? Is that, maybe it's just a stalk with a rock behind it? Just my eyes.
Yeah, I think
We
I
can
see
keep
what
going.
you're seeing. Yeah.
Thought it might be a fan or something, but it's not.
Actually, it does look like a polyopogon, but they're all the polyopogons are kind of brownish, like not healthy.
Another sea cucumber here in the foreground. It's moving a little bit.
So washing its face off.
Oh, and we've got a whip coral too.
Yeah.
Zoom in, Dave.
Maybe it had its head up trying to catch stuff out of the water column. Oh,
That's pretty. That's a different whip than we were seeing before.
it is, and I am not sure what it is.
Bridge nav.
Little sea star on the bottom to the right, or might be a
Can we
brittle
move
star.
another 30 meters to the north, please? Thank you.
I keep waiting for that sea cucumber to like lift off. It keeps lifting its head. I'm like, swim, swim. But it's not going
Get
for
swept away if it comes up.
Yeah,
off the
right.
bottom.
He's like holding on for dear life.
it. It's like, well, you're crazy, lady. No.
All right.
All right,
Thank
moving
you.
on.
I'm going to reset you again.
Go for it.
All sorts of different sizes with these stalk sponges up here. Got some little ones. Looks like we might see a coral in the background, but it's really far back there.
Yeah, it's whipping.
Can we, oh, I don't know if we can
Erica,
zoom
can
there
I
in
borrow
that small
dry
coral on the
erase
bottom.
marker?
Which small coral on the bottom? In the crack?
Yeah, the bottom towards the right.
Can you zoom in, Dave?
Yeah, sorry, in consultation
Sorry,
over here.
Dave. Just looking. Oh, it might be a sponge, actually. Okay, yeah, that white.
Oh, good eye.
Yeah, that's a little different from what we've been seeing. It's another glass sponge. Different formation there.
I'm going to mess with them when they're...
Thank you. I know it's a challenge here with this current.
Is the weather kicking up? What's happening?
I shouldn't...
Not up
Too
here.
much.
It
to
just
us.
feels like we're moving around more.
Oh, from the ship?
Yeah.
Oh,
Okay,
and
we're
another
starting to...
Chrysogorgia
Yeah,
there that I
starting to
missed.
see a little diversity.
Is that a Paramercia over on the left? I mean, the right? The yellow?
Whoa, look at this little skate. There's a skate.
Whereabouts?
Wait, is it? Or is it a sponge?
No,
I think
it's not a... Oh, maybe it's a broken sponge.
I think it's a broken sponge. I was all excited for a
Looks like
second.
a skate. Look at that broken sponge.
Oh, jeez, it's coming after us.
It's coming for you. But do you see that yellow in the upper right?
Upper right of the main video?
Of the main video, yeah. Right.
No.
It's
Zoom in, Dave.
center right now.
It's just some dead thing.
Is it a dead thing?
Yeah.
Aw,
But there's
bummer.
a couple of percentages.
There are some percentages, yeah. Okay, just a dead sponge. It is kind of interesting to see the insides of it.
Yeah.
You want to see the inside of the
No.
dead sponge?
No, we're good. We can keep
All right.
moving.
Maybe in the future when we live underwater, we'll have sponges grow our houses.
Oh, that'd be kind of cool. I don't think I'd want one of the very
Bridge,
clear
bridge
ones, though. I'd want something with a little privacy.
Oh, you saw the article about the underwater?
Yeah, I saw
housing
that.
thing?
Can we add another 30 meters
No.
due north,
It's
please?
called deep,
Thank you.
which
Yeah.
undoubtedly stands for something, but yeah, underwater habitat.
I don't know. Then all you got to do is watch that the video about the what was it, Rayford dolphin or something like that. Like when they accidentally open the hatch when there's a big pressure differential and the guys were squeezed through a little 10-centimeter hole.
Ooh, that sounds horrible.
I don't think I want to live in an underwater thing like that.
Oh, Yikes.
There's like 300 commercial divers that died in the U.S. this year.
Are you serious?
Yeah. They're like having a big conference about it.
Oh my goodness, that's horrible.
Yeah.
Is that just one of those Romella-Gorgia there on the center left? Little fan coral down at the base of that front yellow stalk sponge.
Zoom in, Dave.
Or not quite the base, a little.
Oh, yeah, I didn't even see that. Yep. Yep, that's what it looks like.
It's pretty. Can definitely see the current down here. A lot of stuff moving around.
Yeah, but those tall sponges don't seem to wiggle around too much. They must be strong in the stalk.
They come from strong
What,
stock.
is that what that did sound like a really weird way to say that? Yes. He's strong in the stalk.
Oh, another not alive Faraday, unfortunately. Eventually we're going to find one of these standing tall somewhere. They're probably right over the ridge, just out of sight.
Yep. That's where all the best stuff is.
Just where you can't see it. That's why we
Also,
have eDNA for.
It's also right at the end of dives, if you could just go a little further.
Yeah, sometimes I feel like a little kid, like, just 5 more minutes, 5 more minutes. Can we just
Yeah.
stay a little longer? I want to see the good parts of the bad movie.
You wouldn't be a real scientist if you didn't do that.
That's actually the first thing they teach you. It's like, all right, look, you got to demand more time. There's 3 ways to do it. There's the pleading, the crying, and the yelling.
Oh my goodness. I love that we have these little tiny sponges all the way up to these big ones.
Yeah, so how do sponges reproduce?
Oh,
And just give me a lot of
Branch,
detail, okay?
staff.
give you a lot of detail.
Can you move 30 meters at bearing 325, please?
I want to say they spawn, but that might not be correct.
Maybe they bud.
They might bud. I bet they do bud. Let's see. Many of them are hermaphrodites. Trying to remember
Oh,
here.
another tree. Fallen tree.
Ugh,
Oh,
I don't want to see fallen trees.
look at that. Yeah, it does totally look like a tree, doesn't it?
That one's not nearly as majestic as that first one.
Oh.
It's more of a Charlie Brown tree.
It's still pretty big, though. If we saw it
Yeah.
standing up, we would be oohing and eyeing over it, for sure. Unless we'd seen 100 of them already, and then we might be like, oh, it's another one. A lot of, are those brittle stars or Bersengids on there?
I think they're brittle stars.
Yeah, looks like.
But you're talking to a geologist, so
You want to zoom in, Dave?
Oh yeah, let's see it. It's probably not going to help me though. They look like brittle stars.
Lots of brittle stars. But they're pink ones here, and like the white ones that we saw at Vailulu'u.
Yeah.
Oh, that one almost looks like it's a flower on the right. Like if you
Yes.
look at its central disc, it just looks very flowery to me.
Yeah. Yeah. See that?
Okay, I'm good. Thank you for zooming in on that. It looks a little different than that first one that we looked at. I don't see as many of those kind of chimney type formations that we saw in that first really big one. So it might be a little different, might be a couple of species.
Yeah.
Okay, so refresh my memory on sponge reproduction. So they can reproduce, many can reproduce asexually by that budding, so a piece of it falls off and, or not really falls off, but they can do asexual, but then they're also, most of them are hermaphrodites, so they tend to release their sperm into the water, where it drifts to other sponges, which get it through their pores and fertilize their eggs.
And then, so then they release those fertilized eggs? Or they
Yes.
hold on to them and then when they're born or something, they come out?
I think they wait until they're larvae, so
see,
it
yeah.
develops into larvae that have cilia on it, and then the cilia, the larvae can move around.
Cool.
It does say that some can retain them for quite a while. They can just hold their larvae
Bridge,
till they're a little
staff.
bigger, and others put them into the water column pretty quick.
Wow.
The asexual ones produce buds or gemmules, which are packets of cells of various types, because sponges have different types of cells. So they have like feeding cells, reproduction cells,
Can we move another 30
etc.
meters
So they
at bearing
package all of that
325,
together and
please?
send it off.
Gemmules, that's a cool name.
Gemmules, yeah.
I heard if you put one in a blender, you could just have sponges like they'd all colonate everywhere.
Oh. Crop dusting
Yeah.
with sponges.
I wonder if that's a restoration method. Just chop up some sponges and sprinkle them around with the ROV.
So we're probably doing some of that.
I was going to say.
And Bari.
that
Bari's probably working
hits
on
a little
it.
close to home, actually.
Oh, I see what you're saying.
There's a something.
There's a something?
There's a
Oh,
something.
there is a
Zoom in on the
something.
something.
It's a
Dave?
coral something.
Oh, that's weird. I didn't even see that.
Thank you. Good eye. That one just blends right in.
Ooh, that's different.
It is different. And it might... Oh.
Oh, boy. Oh, now Val and
Gorgonian, I
are-
think.
A Gorgonian. I'll go.
But what kind?
Oh, that's not enough. Jesus.
I'm off my coral page. That is lovely, though.
Oh, I got it.
You got it?
Primnoid Calagorgia crescentus.
Right.
Oh no, looks like a black coral though, huh? Is a primnoid a black coral? Well, the picture I'm looking at looks just like that. That's the problem with these corals.
I know.
There's just too many of them.
There is a surprising number of them.
I'm going with primnoid. Anyone on shore gonna help us here? We're flailing about.
I know, we need our support team.
Steve Oskovich, help me.
It's worked before.
You need a button back there.
Yeah, I know.
Takes a snap and sends it to him.
And I would love like a bright red old style phone, you know, with like the receiver that you
Yeah.
pick up.
Instead of a bat phone, it'd be like a coral phone.
Yeah.
You could probably get ChatGPT to make you an app that does it, though.
True. All right. Well, we got enough imagery.
Yeah.
Wait, you want to? Wait. Can we sample it?
Really?
Just a snip and slurp?
Okay. Are we stopped?
No.
Are we gonna be?
How far off
we're-
are we?
We're tailing behind the ship a fair
All right,
amount.
fine. We'll see the
Oh
next
yeah,
one.
but that
if we
was
can go
by
for it.
natural current. Oh,
Yeah, mostly, but
we can do it.
We can do
Why can't we
it.
do it?
Someone on the chat say it's an umbellapathes.
Umbellapathes.
Umbellapathes, yeah.
Oh, that's above my pay grade.
I don't have the good guides like you do.
Well, really? Mine are not good. I don't see this one.
All right. Esteban, you be my camera guy?
Erica, I've
Zoom out a
stopped
little, Dave.
pump 2, and it's done. So it's
Might
157
have it
meters.
up close and personal too much. We could do it. Of course, the schnozzle's not in a good spot. What canister we need to be on?
Erica, are there anything in
Bridge,
the
bridge nav.
cans already? Slurp jars?
Can we move 30 meters
Yeah, we got
due
something
south,
in 1 and
please?
2, so we can do 3 to 7.
Thank you.
3 or 7?
3 to 7
3
is
to
open.
7. Okay.
And those rocks are still kicking around the flush jar.
Esteban, are you able to come up anymore too? Just preemptively.
I gotta get away from this to get the arm out. Can you push the porch out of Scotia? It's good.
Bob, this wall does seem pretty tall. Pretty
So what are you
close.
saying? We can't do it?
Yeah, probably not.
All right,
Yeah.
we're out.
Okay.
Thanks for trying.
Yeah. A little bit of bobbing up here.
I feel like I've seen a picture of something like this and I cannot find it.
Yep. Pulling up.
You get 7
Not
meters.
zoomed, not zoomed in, right? Yeah. Nope.
Unfortunately not.
Coming up.
All right, don't get crazy there, because you're going to yank me right the hell off of here.
Oh, like that?
Yeah, like you're doing.
Yep. A little bit more slack.
Are you still? Oh, the ship's coming back.
Yeah, I thought I'd try to alleviate our forward motion if possible, but
Give her the beans. Just getting close.
All right, keep pulling up.
Yep.
Just whatever. Just go. Yeah, I can't make headway here. I think we're going to go into tow mode.
All right.
Just let me know.
copy that.
Erica, can you note that in the sea log?
All right, we're off the
Yep.
bottom.
We're over.
Yeah.
And also note that I've started pump 3,
What time did you start pump 3?
2015.
Okay,
Are we
just
nearing
now.
the top of this?
Not really.
Are we on a ridge? Yeah.
So I'm happy to move to the north I'm sorry, the west side of it, if that's going to block that current.
The west side of it.
You said the current's coming from the east
No, to the east.
to the east?
Yeah,
So
look at, here's the rear of the ship, and here's where Atalanta is. It's being blown like, woo!
Okay, so if we move to the east
Yeah,
side of it,
we're
then
already going that way.
All right.
Naturally.
Perfect. Perfect.
That was pump 3.
Pump 3.
Okay. That's sample 215.
Isn't wind directions from where it's coming from and current directions where it's going to, right?
Well, that's confusing. Let's pick a pick a model here, folks.
All right, well, I'm going to wait till you get settled. Too much flailing here.
Yeah.
Bob, what do you want to do for...
I want to just wait till we get settled.
Okay.
And then
It
we'll,
seems like we're Atalanta's pretty stable now. But we can start coming towards you as well.
well, if we're going to, yeah, how far are we
watch-
going to go to get on the east side of that?
Yeah, why don't you move the ship eastward?
Yeah. Bridge nav.
Yeah, I'm just like a, whipping in the wind there.
Yeah.
Can we move 30 meters to the east, please? Thank you.
Guess we see why those big tree corals fell over.
Yep.
Just too much current. It's kind of interesting because most of the surface currents go the opposite direction.
Yeah, it's true.
Well, if you're just tuning in for the first time and asking about our dive location, we're actually at the Malulu Seamount, located within Mudiaba unit of the National Marine Sanctuary of American Samoa. And this seamount is located between Wailu'u Seamount and Rose Atoll. And I think there has only been one brief ROV survey of this site, conducted back in 2017.
That's correct.
And our goal is to study the geologic history of this feature as we survey the biodiversity of this area.
Right now we're battling a very strong current on the seafloor, so we're kind of moving the ship and Atalanta and Hercules around to have the best position to be able to move up the slope, but also get a look at the seafloor. It's kind of one of the inherent complexities of doing work in the deep sea. The challenges aren't just the temperature and the pressure, but also currents and water clarity and all these things combined that make it, you know, tough to do. But of course, the Nautilus and Hercules crew make it look pretty easy.
Sometimes.
And reset the DVL.
Well, we're still seeing these bolosoma sponges here, despite the currents. I think a little Ramilogorgia there on the upper right hand.
Bridge nav.
Now that we're seeing the bottom again.
They don't let me see the chat because sometimes
Can
we
we
get
have
a lot
another
of
30 meters
negative
to the east,
comments in
please?
the peanut gallery.
Yeah.
Yeah, now we're hiding behind the wall.
Is it drop the currents way down?
Yeah, I'm gonna go hands off.
Wow.
Yeah, this is better.
But what about Atalanta? Is it still getting pushed?
Yeah, not nearly as much.
Well, but we're
Well,
moving
yeah,
that
we're
direction.
moving that way.
So what can you tell us about these rocks, Adam?
Well, these rocks are, you know, it's a little hard to tell with these because they've been so heavily encrusted with manganese and iron oxides, but sure looks to me like a wall of pillows.
Can we zoom in on that upper right-hand corner? Is like a little coral just to the right and above the laser pointers.
Right and above, like...
I think it's a coral. It might be a feather star, actually. Never mind.
No, it kind of
What?
looks
Never
like
mind.
a crab.
Can you zoom in, Dave?
We gotta find out what it
It
is.
looks like something.
Definitely is something.
Aha! It is something. It's a crinoid.
It's a crinoid. Just a feather star. We haven't seen too many of those. Oh, there's a little coral below it, though, to the left. It's very tiny. And one to the upper right, too. They're tiny, tiny. You just went past it.
All right.
It's tiny. I'm not even kidding. It's like maybe 3 centimeters. See, right below the laser pointers.
Yeah,
I don't see it.
it's right there.
Might be a hydroid, actually. Very small. See it?
Oh,
Unless
yeah.
It's just marks on the rock.
Holy cow. Oh, my God.
And
Well,
I think there was a stylaster to the upper right, but it's also teeny tiny.
All right.
I mean, the closer you get there, the more stuff you see.
Right next to the laser pointers right now.
Okay, I don't need to see that
I'm not saying you have to. I'm just saying it's there.
one.
There's little things. What's that kind of purpley thing down to the right bottom corner?
Purpley thing? You mean this?
Lilac-y. Yeah. I keep forgetting you have that telestrator over there.
Yeah. It's much easier when you have the telestrator.
Oh, that's a nice one.
You zoom in, Dave, at this little tiny stick thing.
It'll help Megan later. For whoever has to annotate these.
Yeah,
Nice
it's
little-
a coral.
Some feather stars on it.
you come down a little bit?
That's great. Thank you.
How close are we? Oh, you got 10 meters. You got it.
Which, so
I've-
we're,
I just looked at way too many corals trying to find that last one that we wanted to. Don't ask me for a moment.
so up the slope is to your starboard, is that right?
No, we're right at, the wall is, if I face to the,
Yeah, but like not local slope, but the main volcano.
yeah, I think we
slope.
went
Yeah.
off we went off the top of the ridge. It was just on the easterly side.
Okay.
And it looks like Atalanta is at the top. So we're close to the top right here. Like
Okay.
the edge, if I just come up a little bit. I
Yeah,
think we'll
of this little wall, but like the
Yeah.
main direction we're going is you're
Is pretty
crabbing.
much due north.
Yeah, we're crabbing along the ridge.
Yeah.
It'll be like 015 once we start going that way again.
I mean, this is where you want to be anyway, right? Right along the edge.
Yeah. Yeah, there's a little whip coral.
There you go.
Is that a dying sponge?
Dying sponge. Get to see the internal,
We
All
zoom
that
in, Dave?
internal
like
structure.
vascular kind of structure.
It's like its skeleton getting ready for Halloween.
Spooky.
Very cool.
Bridge nav. Can we move the ship 30 meters at bearing 015, please? Thank you.
Yeah, this is much better over here.
Johan, can you show me, zoom out on the high pack a little bit just to see where we are?
Yeah.
Oh boy. We got a long way to go.
Yes, we do.
So I recommend we just keep moving so we can eventually get up to the top of this seamount.
So less smelling the roses.
Less smelling the roses, on the fly identifications.
Hey now, these are something really cool.
They are
But
cool.
I do agree we should keep moving because I really want to see. I think we've kind of maxed out on our species in this zone, so it'd be great to keep moving and see what's waiting for us a little shallower.
All right. You going? Where are
The
you going?
ship's
Point
moving.
3?
015 right now.
Point 3?
Yeah, yeah.
Although, if we see one of those giant sponge trees, we should hit the brakes.
Yeah, sure. We always stop for sponge trees. That's what my bumper sticker says on my car. I brake for sponge trees.
I think that's the end of your tethering.
Right next to the one that says my other car is an ROV.
going on there? The tape's coming off.
I got one that says, I like my control van's Arctic temperatures.
Yeah. Well, it was pretty hot and steamy in here when we came in.
There's a lot of variation in the temperature today, and people have been messing with the air conditioning.
Who's that? Who's doing that?
Not sure. Janek
Oh, that's fun.
texted me about 5 o'clock this morning, saying it was getting warm in here. And I don't know if people took it upon themselves. Both of them were on this morning when I came in, and they were in auto mode, which meant when they got down a degree or two below where they're supposed to be, they were adding heat.
Oh yeah, when I came in, the window was steamy.
Yeah.
Back here.
Somehow they got into heat and cool mode.
Bridge now. Can we move 30 meters to the north, please? Thank you.
It's great now. Not moving along very good though.
No, it's slow to respond.
Yeah, I think Atlanta is still affected by the current.
Totally, it might settle back down another 20 meters.
Well, that would be good, actually, right? Get off the ridge a little bit.
Yeah.
Well, I got a comment and a questions about the sponge explanation that you were that you did on the last few minutes, and it says surprised about the commentary about the sponges, and the question is, how often do we discover new sponge species?
Oh.
There's like 3 questions, and the second one is that, are there any land-based relatives to sea sponges? And then the third one is, do sponges spend their whole life in the same place? And if not, why do they move?
Okay, let me see if I can tackle those. So the first one, how often do we find new sponges?
Yeah.
I would say probably pretty frequently, because one of the challenges, even in the shallow reefs where we have easy access to sponges, we still don't have identifications for most of the sponges. So you almost have to identify some of the sponges, you actually have to look at their spicules, which is the little tiny supporting structures inside the sponges, which are distinct by species. And so that's one way that people classify sponges, is by looking at their internal structures. But to do that, you have to actually take a sample. So we're still constantly discovering new species of sponges. And I think that's probably true in the deep sea too. So we already took, the team before us took a few sponge samples already, and we've taken samples at other sites too. So that's a great question. I'm not aware of any sponges on land. I do think there's maybe some freshwater ones, but not many. They're mostly, I feel like they're mostly marine. And I might be wrong about that freshwater. Might be confusing it with another group. And then what was the last question?
Do sponges spend their whole life in the same place?
Once they settle down, they do. So they can have, when they're larvae, they can move around. But once they kind of set down, they're kind of like corals in that way. Once they set down, they're kind of there.
Okay, and then the last comment is thanks for sharing your adventures.
Yeah, thanks for joining us.
Yeah. No,
Where's that person coming from? Can we tell?
no, we can't tell.
All right. If you write in a comment, tell us where you're from. It's cool to know where people are tuning in from.
All right. Bridge nav.
You know what we haven't seen on this dive?
6-gill shark.
I haven't seen any fish yet.
Octopus.
Oh.
Haven't seen any
30
octopus.
meters
Or
at
an
bearing
octopus. I could go for
030,
either.
please.
Did we
Thank
see
you.
any shrimps?
We have seen a shrimp.
Okay.
One
Yeah.
photobombed right in front
Oh,
of us.
yeah. It was just one.
And we also have seen some squat lobsters.
Yeah, squat lobsters.
In that Christ Gorge today, so that was really nice. I did have a question for whoever said that Megan identified our sparkly cephalopod as an octopus. Did we get a species on that sparkly octopus, or just that it was an octopus?
Hey,
Pelagic.
anybody know what Megan said about the
Pelagic.
octopus?
Pelagic octopus.
Pelagic octopus.
A pelagic
I
octopus.
believe that was what she said.
Yeah, I was wondering because I didn't see a distinction after I thought about it more later that day. I didn't see a distinction for the mantle covering like the squid has. So, and I also didn't see longer tentacles, just the arms. So those are features we should look at when we see another cephalopod. So, but so far that we've only seen two. We saw one in our I think it was our very first dive with this watch that we actually saw bottom. We saw an octopus. And then I think it was on our watch. Maybe it was someone else's. Thought it was on ours. And then we saw that one, the pelagic octopus. And we have seen a lot of squid in the water column, especially when we're up in the shallows at night on our transits. This is interesting. It almost looks like it's a dead bamboo coral that's been eaten, this whip, but maybe not. It seems like it's got knobs there on that stalk in front of us. The white one with the brittle stars at the top of it. And we've got more dead sponge stalks there at the base. And we're kind of understanding why now. And we've got a nice coral here in this towards the center. All right, I can. This one is a branched coral. Thank you. That was good. Got some more smaller sponges coming up here. And it looks like, is that another, oh, just branches. Looking at our fisheye lens, which gives us a little bit of a peek ahead. Thought maybe we had some whip corals, but they're just the stalks of sponges. I think we're starting to really understand why there's so many stalks in this area, because there's a lot of current. So these animals are, benefiting from the current because it probably brings them more food particles, but it also is a challenge for their structural integrity. Got a sea cucumber up there on the left, on the bottom. It's pink, just passing off screen. And it looks like we have another coral coming up ahead on the upper right corner. Sorry, I don't have the Telecaster to zoom you in.
Telestrator.
Bridge,
Telestrator,
bridge
sorry. I've heard it called like 4 different things. Keep forgetting what it is. It's up towards the
Yeah.
center.
Can we move 30 meters at 045, please? Thank you.
Right there with the lasers. Might be the same ones that we've seen recently. It looks like a standing Faraday sponge here. I don't know, it doesn't look very healthy.
Looks like it's about to fall over.
Yeah, but it's closer. We're getting closer. We've got one standing. Now we just need one alive and standing.
Yeah.
Nice big bowl of Selma here. It's doing fine. Looks pretty healthy. Looks like there's a sea cucumber down to the right. Might be a fish, but I think it's a sea cucumber. Bottom right-hand corner.
Yeah. I think they're sorting out positioning, so we're probably not getting any zooms here for a couple minutes.
No worries. Might be 2 sea cucumbers there. Quite a few stalks here, missing their big spongy top. Just
Those
the stalks there.
sponges looks like helium balloons.
Yeah. Yeah. Kind of wonder if like there's any buoyancy to the big head, but I think it's not. It's just mostly water, and then silica, which should it be a little bit, you know, denser than water, and then a strong stalk.
Have a Brisengid sea star there on the right. That pink thing with many arms. So it seems like there's a little more sediment over here. Is that just because it's getting brushed down? Oh, Adam doesn't have his headset on. Never mind. I'll wait for geology questions. Nice whip here. See all of those
Bridge now.
polyps reaching up. Moving up in the water column allows
Can we have
them
the ship
to
move 30
access
meters at bearing
more
030,
food. They're
please?
filter feeding out of the water column.
Thank you.
We have a nice coral there on the left. It might be similar to the one we tried to snip and slurp earlier. Far left center screen. Nice long coral whip there in the upper right-hand corner, along with some bolosoma sponges. Some other stock sponges might be of a different species, different group. And of course, these whips are made up of small individual polyps, so you can...
What was that?
Val's been working it.
Can you not hear me, Bob?
I like it that way.
Ouch, ouch, Bob, ouch. Here I've been describing
Like,
what
man,
we're
I can
seeing
just- it's
down
like-
there.
No backseat drivers.
Well, that's because Adam's back here talking off headset, so he's not tuned in, which means it's been all biology, so it might be a little boring. It looks like we have a coral in the upper right-hand corner. The white formation there. It looks a little different than the other ones that we've seen so far. Maybe a little more fan-shaped. Or is it? No, I think it's another one of those glass sponges we saw earlier. So that very...
Club shaped.
Yeah, so it's more like a vase shape with these kind of protuberances coming off the side. It's kind of pretty. Amazing to think these
Zoom in, Dave.
are made of basically glass. They're silica. There you go. That's lovely. Can see the inner structures kind of woven.
Can you, Esteban, be my camera guy? Try and look down in there.
See if there's any shrimp or anything living inside. These glass
bridge nav.
sponges often have some small crustaceans associated with them.
And I'm pretty sure that's a euplectellid. Corbitellini.
Can we
I
move
know,
30
you know when I'm
meters
saying
north,
it really
please?
slow, I'm reading it right off the page, even
There's-
though I'm trying to sound like
We're
I
trying to
know.
look down the throat of it. If you can
There
tilt
is
down
nothing
more.
wrong with that.
Keep tilting down. We're trying to look down the throat. That's about as tilt downy as we can get.
Okay, I'm preparing for a jump scare. I don't know what's going to come out of this thing.
It's probably just a tiny shrimp if there's anything in there.
Well, it's done a good job keeping its secrets hidden. That's cool.
It's really pretty.
Yeah, really nice.
Little feather star crinoid there on the left. We're up in the center now.
Yeah, there's
Oh, there it is.
Oh,
Yes,
got
we
it.
found one. Nice camera work. Thank you, front row. Now some of these sponges close in, and so the shrimp larvae come in and then get stuck there.
Yeah, there's some like,
that's the Japanese wedding.
yes, exactly. That's what I was thinking. Right? Yeah, they give the sponge. What is the message of that?
This
Well-
is together forever.
Yeah, there's like two. There's usually
Together
a pair.
forever, trapped.
There could be a message there. Let's not dive into that. A bunch of you have spouses. Might not be a good conversation. Oh, look at that.
Oh, that's cute.
You can also see the tiny hair. I think those are tiny. I don't know if that's something else or if that's the hairs on the feeding structures on the sponge there.
Oh, yeah.
See those really
Yeah.
fine hairs.
Right.
So it could be cilia on the sponge, or it could be
Keeping
something else
Dave
kind
busy
of.
with the focus knob.
Yeah.
Could be something else colonizing the inside of the sponge.
All right, I'm happy. Let's
All
move
right.
on.
That's great. Thanks so much. I know it's challenging.
Yep.
While you were off mic, Adam,
Yeah.
Bob just turned off SPL. He didn't want to hear the biology stuff. So maybe you should tell him some rock
I
stuff.
didn't do it on purpose. Bob, you never told me how to do that. How do I turn it off?
Gonna force feed you all biology, sorry.
It's cool. Like at the end of these cruises, I know so much biology. Unfortunately, I don't practice it, so it kind of disappears.
Yeah.
And then I learn
Now
it
we're
all
getting
over
a little
again.
Too far back there. Can drop it down a little.
So I was gonna ask you earlier when you were off headset, like, seems like there's a little more sediment on this side. Is that because we're kind of a little bit in the lee of the current and there's
Yeah.
more ability for it to settle down here?
Falling off
Yeah,
the top.
100%. Like, this is actually one of my favorite things in geology is that when you look at a old ancient sedimentary record,
Mm-hmm.
just by seeing like the kind of sediment, you can tell what the environment was.
Yeah.
So like just as you go out, walk out the beach, you know, right at the shoreline, it's kind of coarse sand. And then you get deeper and it starts to be finer sand, and then even deeper and it turns into mud.
Ridge,
And
ridge
so
now.
looking at the grain size in the sediment, even from ancient records, you can tell what the kind of energy environment was, which
Oh,
was
yeah.
just so
Move
cool.
30 meters at bearing
You can see
330, please.
ripples preserved in ancient sediments that mean that there was current
Thank
and all
you.
that. It's just, it's just like the biology, right? Like everything here is telling a story. You
Yep.
know?
What are these tiger stripes saying?
Yeah, those are ripples from current, and then like a density sorting on the, just like we were on the lee side of
Zoom in,
the
Dave.
Ridge, and we got more sediment. The lee side of these ridges, or the low side of them, is where you can collect the denser.
So it's like a
Sediment.
micro habitat of like what we're looking at right now. So there's like a little lee in these little,
Yeah.
they're almost like little sand dunes if you were a
Ex-
tiny critter.
Exactly the same process.
Very cool.
So you can move sediment with water, you can move sediment with wind, which is where you get sand dunes from on land or on other planets. And when I spent some time down in Antarctica in a place called the Dry Valleys, there's surfaces there that are like millions of years old,
Oh, wow.
and rocks that have like worn smooth just by sand blowing across them for millions of years. Very cool.
Kind of mind-blowing to think about.
Yeah, it's one of the few places on the planet that has you can walk on a surface that has been around for millions of years. Otherwise, it's pretty dynamic, you know, just forests grow and erosion happens and
Soil
mountains
gets
lift up and
deposited.
soil gets
Yeah.
deposited, yeah.
Very cool.
But likewise, these surfaces are millions of years old. They've changed because the manganese crusts have been deposited, but but otherwise they've, you know, been just as they are for millions of years.
Are there any more comments in the chat, Bono?
One of the reasons, just to add on, one of the reasons just to kind of emphasize that these are millions of years old, on the dive a couple years ago, we picked up one of these rocks and peeled off the black manganese crust, and there was a megalodon tooth inside.
What?
Yeah,
Oh, that's crazy.
yeah, so an animal that hasn't existed on the planet for millions of years, we found evidence of it on the surface of one of these seamounts.
Oh, that's very cool.
Yeah, a question from UK says, have we seen any sea pens yet?
Oh, great question. I've been looking for sea pens. You know, we haven't been in too many places with a lot of sediment. Most of the sea pens like to root themselves in the sediment. But no, we have not seen, as far as I know, at least on our watches, a single sea pen.
Gonna do a reset.
Yeah, I haven't seen any, even in the ones I've been watching.
Do you want to keep coming a little west?
Yeah, I think we
Is that
need
what you're
to.
looking
We're
for?
falling off of the top of the thing.
Copy that. Bridge down. Can we have
And
another 30
UK,
meters at bearing
that means
330,
it's like
please?
nighttime there,
Thanks.
right? So it's 7 hours back to the East Coast, or 6 hours, so it's like, it's late night,
Well,
after dark.
If you go with our UTC time, which is also Greenwich time,
Ah,
so almost 9 o'clock
yeah.
Yeah. p.m.
Oh. Yeah, we saw some tiny sea stars and a lot of crinoids. What's the difference between sea stars and crinoids?
Oh boy, you gotta look that direction. That's our
Okay,
biology
yeah,
expert.
okay, let me
Yeah.
focus
Oh,
on
I
this.
tell you, neither of them are rocks.
Let's see. Difference between crinoids and sea stars? Is that was the question?
Yes.
Goodness.
Oh, maybe they're like referring to like the crinoids and the brasinges look really similar.
Yeah, something like that, yeah.
Yeah.
That's a good question. So they are in different classes. So, or not really, yeah. So the crinoids are just these feather stars and also a thing called a sea lily. And they are, I feel like they're older, like they're pretty ancient. They're
Oh my
one of-
gosh, yes. Again, like definitely, you can see them in the rock record. They were known as from the fossil record before they were known from the modern day.
Yeah. And so, I mean, I remember when I used to go fossil hunting along the, what was it, the Falls of the Ohio, I think it's called. It's down in Louisville, Kentucky. You can walk along the riverbank there, and you can actually find crinoids, like the stalk crinoids. You can actually find them right there, just laying there, and so you can go fossil hunting and look for them. So I've been kind of fascinated with crinoids since I was a kid and picked up some of those. So they also, I think one of the interesting things with the crinoids is they can kind of walk around. But it's kind of different. Like with the sea stars, they have this water vascular system that is moving through their arms and
going
kind
up
of
slope.
tube feet,
Yep.
versus the crinoids have these smaller kind of legs underneath them. I'm not explaining this very
I'm with you.
And so I think it's really interesting because you'll see these crinoids, they'll have these kind of smaller appendages on the bottom that they use to grip onto the seafloor or onto a sponge or a coral. And then they'll have their arms up in the air filter feeding with those.
And when they put their arms in the air, they wave them like they just don't care. Is that right?
Bridge now.
It's true.
Okay.
It's really true. They're just right up there. Oh my goodness.
Sorry, I apologize.
Raise
Don't
the roof.
apologize.
Raise the roof.
We're having fun, and that's exactly what science should be. Science should be fun.
30 meters due north, please.
So keep bringing it.
Thank you.
Yeah, so I'm trying to think, Adam, when did crinoids appear in the fossil record? Do you remember? Like, how far back did they go?
Super long ago. I don't know. I mean, after the Cretaceous tertiary mass extinction event. So, you know, 50 million years, something like that.
Oh, wait, I just found an article. 300 million in this
300,
article. That's
great.
kind of mind-boggling. These animals have been around, these types of animals have been around for so long, which is very, very interesting. So that's them. And then the echinoderms include the sea stars, the sea cucumbers, the brittle stars, sea urchins as well. They're a little younger in their evolution. They all seem to have...
Wait, so sea cucumbers are related to sea stars?
Well, yes, they are.
Huh.
And sea urchins. So it's kind of, you know, it's,
Cool.
yeah, but they have very different, you know, lifestyles, which is pretty interesting. So,
It's interesting that you called it lifestyles.
well, you know, sea cucumber, their lifestyles like laying
They're
around
like-
on the bottom, feeding on detritus, versus like some of the sea stars are out there predatory, preying on things.
Yeah.
So,
And what about urchins? Are those predatory?
most of them are herbivores.
Oh,
You could call them predatory because, like, there's issues with urchins.
they're like, can't they like dig into a shell or something?
It's
like
like I'm up on the ridge, you know.
Yeah.
Or is that, I'm thinking of sea
How's
stars.
the
I think
current?
you're thinking of some of the snails
Okay.
and the sea
Okay,
stars.
this is a good spot. Yeah, you're going northerly again, so I think we're good.
Yeah. Okay. Great.
But yeah, so I think the echinoderms are much more diverse.
Yeah, really.
A lot of different functionality. So there's predatory ones, there's herbivores, there's detritivores, versus most of the crinoids are filter feeders. So they're filtering particles out of the water. And so you'll find them in really high current areas, whereas the sea stars have really diversified.
Ooh, I'm looking at the echinoderms, and one of my favorites are like basket stars.
Oh yeah,
Zoom in, Dave.
that coral is nice here on screen. I love basket stars, and I don't think we've seen any yet on this, like not the really frilly, beautiful ones. I'm hoping that we'll see some of those.
Yeah, the first time I ever saw one of those was in the Galapagos, and I was like, oh, I've never seen that. Let's get a sample of that. And I don't think we ever got it off the manipulator while we were on that dive, because they just wrap their
They
tendrils
do.
around everything.
Yeah.
Beautiful.
I'm having such a hard
Candelabra.
time with my corals today. I don't know why. They're just not coming to me. But that's lovely. What about you? Got an identification on that one, Adam?
Well, I said candelabra because that
Yeah,
was the
it's
general
bad.
shape of it, but I don't
Can we have another
know.
30 meters to the north, please? Thank you.
We need the coral hotline. Help us. I wonder if there's anybody that does that, offers that service.
Steve Oskovich.
Yeah, exactly.
We need
Someone
the
on the chat say primnoid.
Oscophone.
Primnoid could
yeah.
be, yeah. Yeah, it definitely looks like a primnoid.
See, it seems like you could have an AI identifying those things for you too, right?
I'd take that. Even if it was right 50% of the time, it'd be doing better than I do.
It would at least give you something to go by, right?
Yeah.
I'm going to go with that primnoid. It looks, we've got some similar ones in our guide, so.
And
Thank
that's what I
you,
thought
commenter.
the really frilly one we saw early on was.
But it had longer, thinner branches,
Yes,
which made me think.
But there's another primnoid that looks like that. But it was a black coral, though, wasn't
I feel
it?
like it was. I feel like that one we saw was a black coral.
Yeah.
It had those very long, kind of bushy formations. So we'll have to have Santiago or Steve look at it another time and tell us what it is. Any more questions from the chat for us? Did I explain enough on the echinoderm, the sea stars versus crinoids?
I think so.
Okay.
Do you want to come back east more?
Yeah, I think you're gonna have to.
Okay.
Let me see if I can get. This is still going up, so.
Yeah. We have this feature in front of us.
And we're now at about 2,595 meters. The range of the 6-gill shark is down to 2,500 meters, so there's still hope.
So
Maybe in 100 meters. I still haven't noticed any fish. Has anyone else noticed any fish?
No.
Here, fish. No octopus yet either.
No.
So just keep an eye out. They might just be hiding. Or it might just be too much down here. Nice Christgorgia here, right in the center. Could have some squat lobsters living in it. Or maybe not. These are just so delicate. When we see them up on deck as samples, they're just so finely branched. Really lovely.
Yeah, so this is the top.
And we're seeing a lot of
Where you
those
are now?
big Bolosoma stalk
Yeah, I think you
sponges
need
up
to go
here.
back east a little bit.
Copy. Go
Like a happy
030.
medium there.
Yeah. Bridge nav.
Bob, did you say we're up on the top of the ridge?
Yeah.
Getting a lot of the current up here.
Well,
So.
not currently.
Can we move 30 meters at bearing 030, please?
But it might explain why there's a little more,
Thank you.
little higher density of our sponges up here, because they want to settle somewhere where they've got good current to bring them fresh food supply.
So it looks like it's coming. The current's coming down slope more. So that's a good thing. We're going into it.
Yeah. Instead of getting pushed around by it.
Cross currents are no good. We don't have much thrust in a lateral way. Because we have the anti-maintenance fence on the one side.
It's
and
very
the small
tall
thrusters.
whippers. Ooh.
I'm going to reset the DVL.
Those little white sea stars are still with us. Little tiny ones down there on the bottom.
Slow up a little bit.
What is that red thing to the right? Is
Shrimp.
that a shrimp?
Shrimp.
Shrimp.
Zoom in, Dave.
Something new. Something mobile. Catch it. No, not really catch it. Catch it on the imagery.
Yeah,
Oh, that's
red
cool.
shrimp.
Hello there, shrimp. I think that might be one of those nematocarcinus again. Those really fine-legged shrimp. Thank you very much. Nice zoom there. Gonna pause the sampler for just a moment.
I think we're in clear water again.
Okay, it's starting to back up. It's a good thing about the current. Kind of helps move any sediment away from us.
Guys, I'm gonna take a quick break. I'll be right back.
Bridge nav. Can we move 30 meters at bearing 015, please? Thank you.
Ooh, this one's really tall and skinny, this stock sponge that just passed underneath us. Bolosoma there. Fauna, what's the meaning of the word malulu?
Oh, good question. Malulu. Only Malu I know. Like, Malu can be like being sheltered or being covered. But Malulu, no.
I wonder where that came from.
But there's a word, someone says Malulu. I know you know that. Malulu. Like, if you say it like that, if you pronounce it like that in Samoan, it's cold.
Oh,
Yeah, like
maybe I'm pronouncing it wrong then. Maybe it should be malulu. Cold, because it's
it's
only
to
2 degrees Celsius
yeah,
down here.
yeah. Okay, probably is Malulu.
Okay.
But Malulu is cold. Like in here. Malulu.
Malulu.
Yeah.
So maybe I'm pronouncing it wrong. Maybe it is cold. It would make sense because we have Vailulu'u, which is active and hot volcano, and this one is an older seamount, cooled off, not hot. I'm not sure where the name came from for this.
Yeah.
These sponges are very close together. And so we're just continuing to climb up um, this ridge along the seamount. We've started at a depth of 3,000 meters and have been continuing to climb up through this dive. So far we're seeing a lot of stock sponges, some additional glass sponges like polyopogon, and some, I'm spacing on the name of the other one that we saw. We've also seen a few different species of corals, some primnoids, some chrysogorgias, a stylaster. We've also seen some different types of echinoderms. We've seen sea cucumbers, we've seen brisingid sea stars, we've seen feather stars, which are crinoids, and we've seen some shrimp, both free-living and one inside of a glass sponge. We've got another sponge here. I don't think we've seen, might be a polyopogon, but a little different there.
Can we move another 30 meters at bearing 015, please?
Can you zoom in, Dave?
Thank you.
You get some sediment again.
Yeah, I'm going to pause this pump 3 for just a moment. You can see those hairy-like projections at the base that hold it on, so very strong adhesive to hold it in all this current. This is probably also glass sponge here, so just get a little zoom on that. That's lovely. Thank you. So just a little different than these stock sponges that are holding up here. You'll also notice a lot of dead stalks that are lying around. So we have seen a number of these stalks still standing. A lot of them have brittle stars on them that are positioning themselves up in the current to take advantage of that. Higher altitude to catch detritus that they feed on out of the water column. Looks like a chrysogorgid there on the lower left, so that's coral. Might have some squat lobsters in it. I don't know that we need to zoom. I think we've seen a couple of those already. We can keep going. So yeah, some nice habitat. We also saw at the beginning of our watch a very large, what looked like a Faraday sponge skeleton that was meters long. So we keep hoping we'll maybe find one of those standing as we move up the slope here. Looks like we have another glass sponge there on the left of center. I don't think we've seen that one before. Is it possible to zoom in?
Kind of bumping the bottom.
Okay.
You still got to pump off?
No, it's paused right now.
Yeah.
Hopefully we'll see another one.
You tilt down and over, center it up. Dave, can you zoom in?
A little bit to the right there.
Point it out again, sorry.
To the right?
Okay. Ah.
There we go. Yeah, there's a lot going there with all this stock sponges and things. So a little different than this species we saw before. It's more smooth on the outside, whereas the other one had all of those protuberances. So I think that's good. It's enough to get a good look at it. Thank you. I've also been noticing we still have these little tiny white sea stars. If you see these really bright white spots on the bottom, there are these little tiny sea stars. So there are some of those Faraday sponges that I was talking about. These also look like they're in the early stages, or maybe late stages of decay there. And a lot of brittle stars and even some feather stars on them. Close-up view of one of them there in the right, lower right. We still have a lot to learn about the ecology of these systems
Gonna
and
reset the DVL.
what causes these sponges to die off. Um, but at least we're getting a glimpse here and seeing a lot of different sizes, which is always a good thing to see in a community, a range of sizes, both which suggests that there's reproduction happening and recruitment coming into the system to help replenish it.
Bridge nav. Can we move 30 at bearing at bearing 030?
And so the one thing we haven't seen on
Thank you.
this dive are any fish, but we have had some really remarkable fish experiences during this expedition, and you can find those highlight reels at nautiluslive.org.
Yo.
I think our favorites are still, we saw a moray eel at 300 meters and some mola molas all the way down to almost 600 meters.
Yeah, shout out to the video crews and those are working on the highlights videos on those.
Yeah.
Like those are really good. I just got a comment from the interactions. Says thank you so much for the interaction. Like my niece came home from school and said that teacher pulled up a few of videos to explain the oceans, and that's awesome. This is from the interaction that they had.
Oh, very nice.
Yeah. And it's from Oklahoma.
Awesome.
Yeah, so for those of you that are tuning in that aren't able to have any register or being able to sign up for our interaction ship to shore, so we got a couple of days there.
030?
Yeah, that's
For
right.
this expedition, yeah.
There's still
Yeah,
there's
we're
still
starting to get-
time to schedule an interaction with us, and I have to
Over on
say
the
it's
other
been
side,
really
yeah.
fun to talk with
You feeling it?
classrooms all over the
I
country.
can see it, but it's not really kicking me around.
Okay, I'll go 060 next.
I know I've talked to students in Guam and Texas, and then we had
Yeah, it's not kicking Atalanta very much.
another group that is more countrywide, so we talked to the other day. So it's been really fun to beam into classrooms and get to answer questions from students. What's your favorite question you've been asked, Fano, in the interactions?
Well, throughout the whole interaction, like yesterday was that question that stuck into my mind. Like, what's the difference between the big Herc and so at least I got some response, great response from our Herc pilots, and
Yeah. Yeah, there's so much technology on this expedition. We have, and on board this ship, so we have these amazing multi-beam sonars that help us map. So we were able to map a previously unmapped and unexplored seamount and then dive on it within hours, which was pretty amazing. We also have the autonomous underwater vehicle Sentry aboard from Woods Hole Oceanographic Institute, and that vehicle is able to create really fine scale maps, and so it's been really great to see the results of that mapping at Wailu'u Seamount. They also were able to put the SAGE instrument on there, which is measuring carbon dioxide, and so it was able to help look for hydrothermal vents within the crater and around the Nafanua cone. So that's been amazing to see. And then here on what we're using right now is the ROV Hercules, along with its partner Atalanta. And so these have amazing cameras on them that you're seeing right now. When that all the speed is beaming to you live. Is that a different another Poliopogon
zoom in,
sponge there? And so we can show you the sponge as we're seeing it.
Can we have the ship move 30 meters at bearing 060?
You can see those
please?
brittle stars on it.
Thank you.
And it takes a whole team to get us to this. So we have our ROV pilot, Bob, Atalanta pilot, Esteban, Johan's charting all the moves and talking to the ship and coordinating, and Dave is zooming in on the camera for us. So thank you, front row, for all of your work to help us explore down here. This is really fabulous. This one's a little more scooped out than the other ones we've seen. Kind of like a bowl. Oh, and we have a shrimp inside, hiding out.
This is one of the 2 sponges that we sampled earlier.
Okay. Are we thinking that's a Poliopogon, Santiago? Or
Yeah,
maybe
it
something
could be.
different? Oh, nice shrimp there, just hanging out.
Oh, speaking of those equipment that have been equipped with the ROV, there's a question from Indonesia says that, what's the
We need
pump
to come up a little.
equipment that needed to be turned on and off so far?
Oh, which reminds me, I need to turn it back on.
Oh, okay.
Thank you. Good prompt there. I forgot when we came off the last time. Whoops. That's a very good question. I was just about to talk about that. I got distracted taking over for Adam here. So the pump that we're talking about is what we call an eDNA sampler. So eDNA is environmental DNA. It's the little bits of genetic information that's floating around in the water. So much like people can shed skin and hair and leave kind of traces of our DNA behind. Animals are doing that in the water column, all the way from including microbes and plants and things as well. So with these pumps, what we're doing is pumping large volumes of water. We're trying to get to 150 liters on each of these pumps. And those are getting filtered across special filters that can trap the DNA particles. And then those are being taken to a lab where they'll be sequenced to see what the DNA pattern is, and then those are matched to a library of known species. So it's really going to help us understand the diversity of these deep sea areas, including things that we can't see. So either things that are off camera or are hiding in the nooks and crannies perhaps, or even down to microbes that are floating around in the water. And so it's a really great tool to help us understand our biodiversity. So we're very excited to have that. There's also the previous mission had samplers on a vehicle called Mesobot, which is a autonomous vehicle that drifts along with the current and looks at the midwater species, the things that are up in the pelagic zone floating around. So another piece of wonderful technology that we have on board that is helping us really understand the ecology of these areas. So great question.
Yeah, again, that's a question from Indonesia.
Thanks for that.
So what's the problem with the sediment?
It can clog up the filter, and
Yeah.
so then we won't be getting as much DNA. It slows down the flow.
So could you like have a coarser filter on the outside or something, or maybe you need like a transmissometer that tells you when there's too much sediment and it pauses the pump automatically.
Oh, that'd be pretty cool. Yeah,
That's a good suggestion.
it's a nice coral here in front of us. Might be another one of those primnoids.
Zoom in, Dave.
Bridge down.
Well, the mapper has one, but the mapper's not
Can we move
connected
30
to
at
a
030,
serial port.
please?
Yeah, the mapper, that's
Thank you.
another piece of the NOAA PMAL, Pacific Marine Environmental Laboratory, has the mapper on board, which is measuring different characteristics in the water column. And so that is also going to help us tell the story, but it's not plugged into the system, so we can't see that until we get it back up on the surface.
And that has both drawbacks and benefits. Like, it'd be nice to see in real time what we're seeing, but to Bob's delight, it cannot produce a ground fault.
This is true. One less thing plugged into the ROV. So,
It could produce a ground fault. I
Yeah.
just wouldn't know it.
Yeah, exactly.
Um, oh, look at this bolosoma here. So
Whoa.
pretty.
I believe sponges are one of the kind of reservoirs of molecules that might have human health applications.
Yeah, this is true. So a lot of sponges and corals have compounds in them that help them avoid predation or help them with their ecological functions. And some of those things have actually proven to be really helpful
Yeah,
in
zip
creating new drugs to combat
Zipping.
things like AIDS and cancer. So, you know, these creatures down here in the deep sea, so far down, might actually be an option for helping us battle human diseases. And there's a lot of folks working on that, looking at the different compounds in marine animals to see what their applicability is for other uses.
Yeah, it's just one of the reasons that it's valuable to explore these environments, to discover new species and to protect these environments and have it, you know, marine sanctuaries and the like to ensure that that we keep these areas pristine enough to be able to explore them and find all the you know, great
Chemicals that are
chemicals
being produced,
and kind
yeah.
of what do they call it? The services, the
ecosystem services
ecosystem
that are provided
services.
by
Yeah,
these.
exactly.
Yeah.
And that's, you know, at least in my view, service to the ocean, service to the planet, just part of the system, not service to humans.
Yeah. So also the ecological functions then. So
Can you
yeah, we
zoom
talk
in, Dave?
about kind of ecosystem functions, which are kind of the things that they're doing, and then the services that they provide to people. Oh, and nice combination here of Christgorgia and a primnoid.
Bridge nav.
I don't know how that brittle star managed to climb up that very, very fine
Can we
Christgorgia.
move 30 meters
They're
to
so
the north,
dainty.
please?
He
But
would be
it's
really good
Thank you.
rock climbers, though.
Oh my gosh, yeah. Hey, have you guys seen that sport that's like speed climbing?
No.
Yeah.
That's wild. They just like zip up a wall and then hit a buzzer kind of thing. It's an Olympic sport? Is it an Olympic
I
sport?
don't think it's Olympic.
I think it made it to the Olympics this last
This last
summer. Yeah.
one? Nice.
I didn't watch any, but so I've heard.
Nice. But yeah, Adam, going back to what you were talking about, that's exactly why places like the National Marine Sanctuaries are important. So they help protect the seafloor here. And this particular unit is also, is that a sea star? Big fat sea star there on the left.
Mm-hmm.
I shouldn't say fat. It's just very.
Plump? What,
I wasn't
what
going
can
to
you
say that either. It's just it's
thick with two C's?
Body shaming.
it's definitely got a thicker body than the other ones that we've looked at.
You zoom in, Dave.
I don't think we've seen this species yet on this dive, so just want to get a nice zoom. Thank you.
Without this pesky old
I know
sponge.
that sponge sock is right in the way.
It looks a little,
This sponge,
gooier?
it almost looks like a glass sponge, but it's
yeah, it
a
does.
sea star.
Oh,
Fascinating.
It does look like it's
We're
like
gonna
just
pause the
lying
pump for
down
a
with a
moment.
big belly. It's like, oh, I ate all of that sponge and now I just gotta lie down.
Oh, you can kind of see its little tube arms underneath. So echinoderms have this water vascular system. So they shift water around to all of these little tube arms that are underneath there. And they can like move those along to help make them walk across, or they can use it to kind of suction and open up things like snails and things. So also interestingly, sea stars put their stomachs outside to eat.
Hmm.
They
Oh.
digest kind of outside of their body and suck in the nutrients rather than pulling the food into them. They're fascinating.
So could be of the family Goniastridae. Is that right? Is that a
Sounds
family? Okay,
sounds
I'm looking through my
possible.
pictures here. Let's see what we can find.
Okay, we gotta get moving.
All right,
Perfectly fine. Thanks so much for taking some time to zoom into that.
Hippasteria, perhaps.
Oh, I think I found it. I think it's a Pteraster reticulatus, or related to that. So, Pterasteridae, with a P, like pterodactyl.
Oh, all right. Good with that.
Cool. Okay, I'm restarting pump 3.
What is that yellow thing down there? Is that a dead sponge or
I don't
a
know.
coral? It looks like dead sponge to me.
Zoom in, Dave.
Dead sponge.
You. Moving on, Dave.
It looks like that one we saw earlier, the white one with the protuberances on the outside.
Yes, which I remembered the name of briefly, and then
I'm the same way. It's like,
This is taking us off the ridge again.
euplectellid.
Bridge nav. Can we move 30 meters at bearing 350, please? Thank you.
We're not there yet. When's this dive ending?
is that
On our
a
watch?
sea cucumber or a sponge
in, Dave.
there to the right? Looks like a sponge.
So that one is on the seafloor in a smooth tube. What do we got?
It's got holes in it.
It's got holes, but it doesn't have any protuberances.
Gonna reset the DVL.
Roger. You play camera guy? There's something living in there. Sorry. I don't think we can look down in there.
If I can pan down and see the whole thing, or yeah, there we go. You got it.
So you can see it's almost like a woven cage, and some of these are encased across the top as well. So that's what we were talking about before, is the shrimp can kind of recruit into them as larvae, and then as they grow, they can't get out. But that one looked like it was open on the top. So
We're coming up some more.
Yep.
Coming up. Coming up.
Nice tall coral whip there.
We gotta get moving here. We can't stop to smell the roses.
No worries. Haven't seen anything new lately, so.
We're right on the ridge here.
I think Megan was commenting at the very, very start of this dive. It's kind of nice for annotation because there's things to see, but they're nice and spread out and not coming too quickly at you. It's kind of nice just to take it all in.
Bridge now.
I'm going to go with yyyy-mm-dd for that.
Can we move
Spun.
30
Okay.
meters at bearing 045, please? Thank you.
It's a nice coral over there on the left that we just passed. It's kind of big and branchy. A little larger than the ones we've seen so far. Looks like another primnoid there passing. Lots of stalk sponges. Anything you can tell us about the geology up here? It seems a lot smoother, not like the bumpier stuff we were seeing. Sir,
Yeah, there's a little change in character. Not quite sure what to make of that, but maybe, different eruption rates produce lava of different types. So at slow eruption rates, you make pillows. At faster eruption rates, you make sheets. Or maybe we're looking at part of the volcano where the eruptions were a little more vigorous. Also, don't necessarily want to confuse volcanic with kind of secondary features like collapses and the like, which once they're covered with this crust is pretty hard to tell the difference.
and we're now approaching 2,500 meters on our climb up this ridge. Where's the top at? Is it 2250 about?
I thought it was like 2450.
Yeah, 2415 is the
15.
Okay.
shallowest.
I think we've got a nice Chrysogorgia just to the left. I think we just moved it out of camera.
It's gonna stir up dirt if I.
No worries, we can keep moving forward. I think we've
So
seen them
that's
before.
that's the thing you got to think about, especially
know,
if we
I'm
have to back up.
trying.
Backing up is the really bad thing. So
Mostly just explaining for anybody watching so they know what we just
slamming
saw.
on the brakes is bad.
Gonna reset the DVL.
Sometimes it just takes me a minute to process what I'm seeing, and it's off camera by then, but we don't have to stop for that.
Bridge nav.
Actually, we made really good time in this watch. We covered quite a bit of distance. That's good. Nice driving, front row.
Can we move 30 meters at bearing 030, please? Thank you.
So despite being up on the top of the ridge, we're not getting a lot of current.
Huh.
Yeah, Atalanta keeps coming.
Yeah, it's almost back to where it belongs.
Right.
So maybe it was a kind of different layers of current. I know our ADCP is
I think it's like really affected by the geography.
The-
It's like it
okay.
gets channeled into the places and, gets rushing through.
Okay.
The Bernoulli effect.
Yeah, probably, right?
Yeah. Good old Bernoulli.
Bernoulli or Venturi?
Bernoulli in this case.
Yeah.
Venturi is...
For wind.
Really? I thought Venturi was...
I was thinking that was more like centrifugal
Yeah.
force.
So Venturi tunnels can refer to acceleration when you change the diameter or the area on which a fluid is passing through. And
What, isn't that Bernoulli as well?
Yeah.
That's
Bernoulli
why
is
I
more
asked.
about
Yeah.
pressure differences. So, yeah.
Hey, ChatGPT.
Write me a term paper on the difference between Bernoulli and Venturi.
Compare and contrast.
It's crazy how you can do that now. You can just type in a question and you get an answer.
Well, or you can just say it.
I mean, it used to be you had to buy that stuff from the smart kid.
Yeah.
Oh, goodness.
How much did you charge?
How much did I pay?
We're talking about some of the new AI models costing up to $2,000 a month.
What?
Ooh.
Yeah, for the ones coming up.
Oh, man.
But, you know, if you're replacing an employee, $2,000 a month is pretty cheap.
Yeah, but if you just want to use it to do
Casually?
something fun, then
Yeah.
you can't afford it.
I think what's more frightening is the energy cost on AI. And we don't really
yeah.
think about it. And so one of the things I find interesting is each AI search takes an incredible amount of energy. And
Not
we're
all
not really
necessarily, though, because another thing they're doing is optimizing and
That's
using
true.
smaller models.
That's
So
true.
I run stuff locally, and, you know, I mean, it's still, you can tell, like, it taxes the GPU,
Bridge
Mhm.
now.
but it's more efficient than
30
using
meters
the,
at bearing 030.
the really big models.
So
Thank you.
some places use deep sea water for cooling.
Mm-hmm.
I just don't know if there's a real benefit in terms of like the energy it costs to pump it up to the surface as opposed to
Can you
other
zoom in, Dave?
methods of cooling.
Are these primnoids again here on the right? And then a chrysogorgia further forward.
Down. And starboard. Down.
There it is.
Ooh. Hey Dave, are you willing to give the cinema cam another shot on that monitor?